What's going on, guys? Aruj again, and today we're talking about meiosis, not to be confused with mitosis. The big idea for meiosis, I want you to think of two big concepts as I explain this. The first thing I want you to think of is a shuffle. We're going to take some genetics, we're going to take your chromosomes, and we're going to shuffle them, okay? The second big idea I want you to think about is a lessening or a reduction. So if I take this deck of cards and I divide it in half, each pile is going to have less than the original. And if I divide that one more time, I'm going to end up with 4 different decks of cards. Each deck is going to have less than the original deck of cards, okay? So the two big concepts I want you to think of in meiosis is a shuffle and a lessening or a reduction, okay? Now, let me go and explain what these mean. So for meiosis, what this process is, it's the formation of sex cells, okay? So when we talk about mitosis, mitosis was the formation of body cells or somatic cells. Meiosis, we're going to produce sex cells, either sperm or egg. Where do we get the name meiosis from? Well, it's Greek, and it actually means a lessening or a reduction, okay? It's also called reduction division. And again, like I said, it's the process of making gametes or sex cells. Each cell is going to be haploid. And if you remember from the last video, haploid means there's half the number of chromosomes, okay? Each gamete is a haploid or has half the number of chromosomes. In humans, that number is 23, okay? Each time meiosis occurs, there's going to be 4 sex cells produced, 4 gametes. If you remember to mitosis, there was only 2 somatic cells, okay? Each cell or each gamete is genetically different. And I'm going to talk about a little bit later why that's important. And there's 2 divisions. If you remember mitosis, we had prophase, metaphase, anaphase, and telophase. In meiosis, it's called meiosis 1. There's 1 division. And we call that prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. Those 2 cells are going to go through a second division, and we call that meiosis 2. Now, why do we put these numbers here? Because they're a little bit different. There's a few things different between each division, and I'll show you guys that in one moment. Go ahead and put this on pause and make sure you guys take note of everything on this slide. And again, the whole idea for meiosis, for the lessening part anyways, is we're going to go from a diploid cell, 2N, to a haploid, or N, okay? And that's the reduction right there. So here's to kind of give you guys a visual. We have meiosis 1, division 1. We're going to start with 1 cell. Pretend this box is 1 cell. That is going to divide to 2 cells. And then we're going to divide that once more, okay? So that's going to be the second division. At the end of meiosis 1, we have 2 cells. Those 2 are going to go through meiosis 2, and then we're going to end up with 4 cells total. So there's 2 divisions. Just like I showed you, that deck of cards, we split it twice. So some key parts in meiosis 1. Okay, these are the main differences than mitosis. Prophase 1. Prophase 1, we have what's called crossing over. What that means is homologous chromosomes, okay? They're going to pair up with each other, and it looks like they're going to play footsies, right? So these 2 homologous chromosomes are wrapped around, and these 2 parts of the homologous chromosomes are wrapped around. They're going to wrap up so tight, this is where the shuffling comes in. They're actually going to exchange bits of genetics. So now this chromosome here actually has bits of this one, and this one has bits of the other one. So now we shuffled those genetics, okay? Crossing over is the exchange of DNA from the homologous chromosomes, okay? So if this is chromosome 1, it's going to pair up with chromosome 1. If it happens to be chromosome 2, it's going to pair up with the homologous chromosome 2, okay? And another name for this is genetic recombination. This increases variation. It makes traits different, and I'll explain that again in just one second. And again, that's the shuffling right there. Next, we go to metaphase 1. In metaphase 1, this is different than mitosis. In metaphase 1, you see that the homologous chromosomes line up in pairs. If you recall mitosis, let's say this was the cell in mitosis, when those chromosomes lined up, they lined up in a single file. This does not happen in meiosis, okay? That does not happen in meiosis, not yet anyways. They line up in pairs. So when those centrosomes separate, they're going to pull the whole entire chromosome apart, okay? And that leads us to anaphase 1. The duplicated chromosomes are pulled apart. So it's the whole one, not the sister chromatids, okay? Go ahead and take note of everything on this slide as well. And now we go to mitosis 2, the key parts. There's now 2 cells. Each one has 23 duplicated chromosomes. Okay, if I go back to this page, see each chromosome has a duplicate copy. It has the sister chromatid attached. Meiosis 2 is identical to mitosis, but there's 4 cells produced, and each one is genetically different, okay? We're going to end up with 4 cells, and they're sex cells that are different. So if we look here, we can see that there's crossing over, metaphase 1, anaphase 1, and that whole thing was meiosis 1. Now we ended up with 2 cells. Those 2 cells undergo meiosis 2, and this part is identical to mitosis. And when these 2 cells split apart, we end up with 1, 2, 3, and 4. Okay? One more time. Homologous chromosomes line up. Whole chromosomes are pulled apart. Meiosis 1 is complete. These 2 cells go through meiosis 2, which is identical to mitosis. Okay? There you go. So, meiosis 1, prophase 1, crossing over occurs. Metaphase 1, homologous chromosomes line up in pairs. And anaphase 1, the whole chromosome is pulled apart. It's going to end with 2 cells. We go from 2N to N. Meiosis 2, it starts with 2 cells. It's identical to mitosis, and we end with 4 gametes, and each one is genetically different because of crossing over that occurs in prophase 1. Okay? So, again, crossing over is the shuffle of genetics. It's the shuffling of genes between the homologous chromosomes. Allocening, that was division 1 and division 2. And we ended up with 4 different cells, okay? That's the big idea for meiosis. Now, why do we have the shuffle? Why is crossing over in prophase 1 so important? Well, this right here is a picture of me and 2 of my siblings. These are 2 of my brothers. And if you look at this picture, you could tell that we are similar. We have traits that are in common, but we are genetically different. Why? Why is that important that my siblings and I are genetically different? How come we're not like this? Identical. Why has nature in biology made it so that way we're all different and not identical? Or why has nature made it that we're all not the same? How come we're all, every person, how come every person is not a genetic clone? Well, here's why. If we are all the same, if everyone is genetically the same, then that means we have the same strengths. If we share the same strengths, that means we have the same vulnerabilities and weaknesses. So if that population of everybody that is the same is exposed to that vulnerability or that weakness, that population is going to die. That population, that species is going to go extinct, okay? That's if we all have the same traits. If we have different traits, then we have different vulnerabilities. That means if we're exposed to a disaster, it's more likely that some members of that species will survive and reproduce, okay? Survival and reproduction is the foundation of biology. Now, why do we have the reduction? Why did we have the lessening of chromosomes? Well, I covered this in the last video. Remember, gametes have to have 23 chromosomes. Once the sperm and egg cell with 23 chromosomes that are haploid, once that egg cell is fertilized, that becomes a diploid and has 46 chromosomes. We call that the zygote. That zygote now has the correct number of chromosomes. So we have to have that lessening, that reduction in chromosome number, okay? That's it for today, folks, and I'll catch y'all next time. Peace.